Thank you. So good morning, everybody, or good afternoon. Um, the title of this talk is Inflammation in the COVID-19 Airway is due to CFTR signaling by the SARS-CoV spike protein. Since we've all been living through the COVID pandemic, a lot of this will not be new to you, but CFTR may be new. And it's the name of a protein mutation which causes a disease called cystic fibrosis. This is something we work on. It's hard to remember actually the name of this protein because the people who were studying it in the beginning at the Hospital for Sick Kids in Toronto had no idea what it did. But they were listening to a rock station, CFTR, Canadian Frequency Toronto, 680 on the AM dial, and they decided to call it CFTR. Actually, um, that's the way I remember it. And it's a way of sort of giving information about what people were doing in the old days when they didn't know what they were exactly doing. But in any event, the protein itself is really critical, as are many of the proteins or genes that are found in rare diseases because they're rare because it's difficult to live without them. We are joined by colleagues at USAMRIID, the United States Army Medical Research Institute of Infectious Diseases, where the very worst viruses are actually studied. And we were able to, so this is a collaboration. And the paper that much of the data I'm going to talk about was recently published in Scientific Reports. Hung Kawi et al. Down below at the bottom. If we can go to the next slide. So the goals of this project were to understand, first of all, the mechanism by which pro-inflammatory cytokine storm in the COVID-19 airway occurred. It's essentially lethal. And second, to find a therapeutic strategy to suppress the storm. And by the way, if you see the spike trimer moving, it's supposed to be moving. It's not your eyes. Next slide. So firstly, we developed an in vitro assay for spike protein binding to ACE2. And then we looked for candidate drugs that could block the interaction. And finally, we found that cardiac glycosides such as digitoxin, digoxin, or wabane, these are all cardiac glycosides, were potent inhibitors of spike ACE2 binding. So after going through a lot of studies that were looking at the binding, we asked the question whether any of those drugs, which are actually approved around the world and licensed, digoxin is licensed here in the United States, uh, actually interfered with infection. So on the next slide, this is basically a slide that shows that the infectivity of native virus is inhibited by digitoxin and other cardiac glycosides in human lung cells, the A549 cell, very typical. So across the top is a series, well, this is a series of dose response curves. The x-axis concentration of drug, the y-axis in green, inhibitory values, K1.5 values, essentially EC50 values, for all three of the cardiac glycoside drugs, and they're in concentration ranges used to treat heart failure, interestingly. The red colors is cytotoxicity. It's about tenfold greater than the respective inhibitory EC50 values, which means that you can use these drugs without getting into inhibiting the cells. The two boxes below the genins are the steroid nucleuses that go with these drugs minus the sugars, and they're not very effective. And that was published in Scientific Reports in 2021. So we were very happy to see that we could actually transmit some in vitro experiment into something that was approximating in vivo. Next slide, please. So after giving you, there's additional preliminary information about CFTR, which I want to emphasize. It's a gene responsible for the control of inflammation in the rare genetic disease, cystic fibrosis. And as we will see down in the next few slides, essential for control of inflammation in the COVID-19 airway. Next slide. So I'm just going to read the first part because it says what I want to say, and then we'll go on. Early in the COVID-19 pandemic, it was found that the lethal cytokine storm was due to activation of the TNF alpha NF kappa B pathway. But how that happened, no one knew. In the middle of this slide, you can see a signaling pathway which starts with, at the top, TNF alpha, which binds then to a TNF receptor in the membrane of the cell. And there it actually connects up to the first intracellular adapter, a molecule called TRAD. The DD stands for death domain, very unnerving. That molecule is a critical director to take all those signals from TNF alpha to the IKK zone that then binds to or phosphorylates I kappa B alpha, releases NF kappa B, which moves into the nucleus and generates all of these cytokines, IL-6, IL-8, TNF alpha, and a bunch as messenger RNAs, which are then synthesized and secreted into the airway and into the rest of the body, actually, in the context of a cytokine storm. In studying this situation with cystic fibrosis, we found that there were some, looking at countervailing candidate drugs like digitoxin. We've already shown you that they interfere, by the way, with the infection. We found that digoxin, Wabane, basically block that process. You can see the little in red letters and little block, a little T junction, showing that when you make a binary complex between TNF alpha and TNF receptor 1 and it wants to bind TRAD, it can't. So that is, the first experiments were just done in plain old HeLa cells, that was actually demonstrated where the problem lay. One function of CFTR is to block that molecule, TRAD, and thereby inhibit the cytokine storm in the CF airway. But in fact, since the digitoxin and the CFTR are all working in the same place, essentially the drugs are phenocopying or mimicking the CFTR, we hypothesized that the COVID-19 cytokine storm was due to loss of CFTR, and that cardiac glycosides, which would basically mimic CFTR, might be therapeutic. Next slide. So the first question was, does spike protein exposure reduce CFTR? The way we actually did this experiment, we used a human organoid lung preparation prepared at the air-water interface. You put some basal cells, which we got from a colleague, into this air-water interface. And over the course of 28 days of farming, you grow a beautiful epithelium. You can see goblet cells and ciliary cells and other sorts of cells verified by immunofluorescence below. So on the right-hand side, we are using Western blot analysis of spike protein effects on levels of CFTR. Along the horizontal axis, we're increasing the concentration in the first upper column, A, ancestral or original, the Wuhan version of the spike protein, and it causes a significant dose-dependent loss of CFTR. We then looked at a much more biologically potent strain. This is the South African beta strain, much more potent. It turned out as a virus to cause COVID, but much more potent at reducing CFTR. You can see the difference in the bar graphs and in the Western blots. So yes, we concluded spike protein exposure does reduce CFTR levels. Next slide, please. Next question was, does spike protein activate pro-inflammatory NF-kappa B signaling? Well, in the graph in the picture that I showed of the signaling pathways, there's a whole series of proteins starting from the top, TNF alpha and TRAD and so forth, which get activated if CFTR is lost. Well, as you increase the concentration of the spike protein, again, we're using the Wuhan level, the strain here, all of those red arrows on the left actually show places where increases is happening. And if you look over, you'll see, yes, TRED is actually increasing. And in the end, IL-8, which is one of the cytokines we measured, is also increasing. So yes, the spike protein activates pro-inflammatory NF-kappa B signaling. Next slide. Question at that point, and this is where our USAMRIID colleagues came in handy, do the cardiac glycosides protect lung epithelial cells from CFTR loss by native virus? On the left-hand side, you'll see a Western blot. Various things are added. Right in the middle, there where it says virus only, you'll see that the level of intensity of that CFTR band, in this case it's the dimer, is reduced substantially compared with the medium control. And that digitoxin and Wabane raise the levels. A bar graph basically shows very similar things. These are all based on 4 independent experiments. And the real situation here is that it's all very well for us to actually do experiments with spike proteins, but it's very important to verify that this is happening in real viruses, in real disease. So this was something that we really were interested to show and to validate. We'll go on to the next slide, please. So your question was, how does spike only, which we know only bind to ACE2, yet it also seems to affect CFTR levels? How could that be? One possibility was that maybe the CFTR existed bound to ACE2 in the membrane. Actually, CFTR is bound to a lot of things in the membrane. Well, it turns out, yes, it's bound to ACE2. The left-hand side of this figure shows that, in fact, CFTR and ACE2 actually are found both in the basal cells that we make the differentiated epithelium from, and in the differentiated epithelium. CFTR is there, ACE2, and actually everything else you need to actually get a nice infection going, as long as you can call an infection nice. On the right-hand side, it shows that if you take an antibody against ACE2 and immunoprecipitate it, and then test that immunoprecipitate for bound CFTR, sure enough, you can find plenty of CFTR. Those are 2 duplicate experiments. The NRS is just simply normal rat serum. You can also find immunoprecipitated CFTR in just the basal cells. So it's basically accessible in all the different parts of the epithelium. And then, next slide. I want to pause here to remind or to emphasize to everyone, CFTR is a cyclic AMP activated chloride channel. That channel is critical for keeping the airway hydrated. When the channel isn't there, the airway dries out and inflammation occurs. But in this particular case, here we have the CFTR protein in the cell surface. Up is airway, down is the interior of the cell. Norepinephrine or epinephrine can bind to a protein that actually does bind, another protein that binds the CFTR called the beta-2 adrenergic receptor. Generates cyclic AMP, protein kinase A is activated, and it then labels the phosphorylates, the CFTR, and the channel activity is activated. You can see a chloride molecule moving out. Why do I show this? Well, you know, if the protein is being lost, you expect that the channel activity would also be lost. It's sort of a test. Is it really true that the spike protein does remove everything associated with the CFTR? So let's look at the next slide. And on the right-hand side is a classical Using chamber experiment, essentially. That's what they call it. But we could actually look over the horizontal axis on the left-hand side is time course, and the vertical axis is the activity of transport of chloride, actually from one side of the air-liquid interface to the air side. And if you add some IBMX or something to stabilize the cyclic AMP, the green, the channel actually opens up and you can see a very nice signal. And if you add an inhibitor of CFTR, it will go down. But if you add spike protein, then you lose about 50% of the signal in this experiment. Spike in this case is Wuhan, so it's even the least potent of these. But if you add Wabane or digitoxin or digoxin to this cell culture, it's culturing over the course of 24 hours, of course, the spike effect is lost. On the right-hand side, you can see that story with the bar graphs, but just below it, where it says CFTR, you can see that when you've added spike, you even get less CFTR. So the spike protein causes loss of the CFTR protein in the same system, and the CFTR protein loss is rescued by treatment with of the epithelia with all three cardiac glycosides. So the loss of activity, channel activity, loss of protein, all occur at the same time. That means we feel we robustly address that concept that the spike protein or the virus, for that matter, has reduced CFTR levels. Next slide. So our conclusions are that the data support the hypothesis that the COVID-19 cytokine storm is due to loss of CFTR. Second, the loss of CFTR activates TRAD to drive TNF-alpha and NF-kappa B signaling. Digitoxin and digoxin and Wabane may be therapeutic by blocking, first of all, by blocking infection, by inhibiting the virus from binding to ACE2, and second, by inhibiting inflammation, by inhibiting TRAD and thus substituting for the lost CFTR. We suggest that the cardiac glycoside advantage is that it is one family of drugs with two simultaneous candidate therapeutic activities for COVID-19. Next slide. We have to have these acknowledgements, partly our lab at Uniform Services, and also the Herbert and Flores lab at USAMRIID. And we also have to have a Department of Defense disclaimer, which talks about the fact that we're not trying to sell anybody anything. And we thank you for that. Next slide. Yes, thank you. And I look forward to any questions that might be there later in the presentation.